Hello, good evening. Good evening, Asraful. Good evening, Jagadish. So it's not good afternoon, it's good evening. So welcome back. So we are into a new chapter today. So today we are going to have a sneak peek into the world of biological classification. So thank you all for everyone joining me. Good evening, Navneet. Okay. So when you hear the term biological classification, it's pretty scary, right? Aman Verma. Okay. So hello, good evening. Okay. So today we're talking about class 11 topic, and that is biological classification. So before we actually go into our topic, that is biological classification, so let me tell you, today is 21st of November, so we are almost on the 325th day of the year, so we still have hardly 40 days to say goodbye to 2020. So 2020 was tough for all of us, right? For every one of us. So it was not good for even one of us, so all of us suffered a lot, but still, let's have at least a good ending in 2020. So for those of you who your target is NEET, so let me tell you, on a serious note, you don't have much time. So concentrate. Good evening, Priyanka. So concentrate and stay focused. Okay, so don't waste time. So 2020 will be gone in a flash of time. Okay, and then it's 2021. So you have other things to look forward. So pay attention. So you just have 40 days for 2020 to end. So let's say a happy goodbye, though it was not a good start. Fine. Okay, so now talking about biological classification. So hello, it says sports talk, so I don't know what's your name. Hello, fine. So when I say biological classification, so this is one of the most scary topics in class 11, right? You agree, right? So all of you are very scared because it has a lot of examples, and the examples are not even familiar to us. So it is all microscopic organisms. So you don't feel like reading biological classification, right? So the topics that are interesting in class 11 are basically your physiology. So all of us love physiology because you don't want to become doctors. So you want to know more about your body, so your digestion, respiration, excretion, etc. So apart from that, your cell topics are interesting. The cell, the unit of life, cell cycle, cell division. Hello, Vishal. Okay, but this type of classification things are pretty boring and pretty confusing, and it's a big headache, right? Yes, it was for me too when I was also preparing for NEET, but now it is not, because you understand, you analyze, you tend to read the logic behind it, right? So we'll go slowly. So we'll take around 4-5 lectures for biological classification, and slowly and gradually we'll grab the essence of it. Okay, it's not tough, it's pretty easy, so we can make things easier as well. So that is the challenge, right? So everything is tough around us, but how smart we are. So it's always put like study smart, not hard. So that's what we're going to do. We're going to study smart and not really hard. Okay, so as quick as we can, as fast as we can, we'll try to finish biological classification with a lot of interest and with a lot of tricks. Okay, so hello Gopal, thank you for joining. Okay, so you're from Hindi medium, Aman, but you like attending my class. Thank you so much. Okay, so if you don't understand any point, so stop me right there, I will explain it again. Okay, fine. So biological classification, we'll make it interesting, so all the examples will be easy as well, but it's not that I am going to give you a magic. Okay, so magic will come if you believe in magic. Okay, so magic does not just come out like that. So we are not living in Howard, right? So it's not Harry Potter, so that I can give you a magic and in a day everything will enter into your mind. No, magic will happen gradually. So as your belief in magic happens, so you will see the result. Okay, so the more you understand, the more you revise, so biological classification will become interesting as well. Okay, so now going forward, so let's see. Okay, so before we actually go forward, it's again a gentle reminder. So there is a coupon code for you, that is KR10. So please download the eCareerPoint app. So in this eCareerPoint app, you can find my presentation, so in the PDF form. So if you are going to revise, so it will be easy for you. So if at all you have to purchase any course or any product, so use my coupon code, but it's valid only till 30th November. So we don't have much time. So hurry up, it's just 9 days left. Okay, fine. So now, actually going to the topic of biological classification. So what it is all about. So what are we going to talk? I said 4-5 lectures. So what are we going to talk in 4-5 lectures of biological classification? It is not easy for me to explain as well. It is not easy for you to listen. But still, something is holding us back that we have to learn. So that thing that is holding us back will definitely make it interesting as well. So first believe and then enter inside. Okay, so if you're coming inside with a mindset, oh, it's going to be boring and it's a tough topic, but still it has weightage, I have no option. No, you can't do anything. So as your belief goes, the magic will also come. Okay, so please trust and join in. Okay, fine. So we are going to talk about the kingdom of classification. why classification is actually done. Fine. And then we are going to talk about each of the 5 kingdoms. Hello Rohit sir. Okay, so kingdom Monera, kingdom Protista. So you would have heard about this, right? So kingdom Fungi, kingdom Plantae, kingdom Animalia, and finally we'll end the lecture with viruses, viroids, and lichens. So you can ask me, ma'am, kingdom Plantae, kingdom Animalia are separate topics as well. Yes, but today in this chapter I will just introduce you the kingdom, plant kingdom and animal kingdom, but will not go in much detail about these two. Okay, so it will be just an introduction. So this virus, viroids, and lichens have not been fitted into any of the kingdom because it has a very distinct characteristic. It's very unique from the other things. So it is a separate one. So we'll talk about that as well. So today let's go forward with why do you need the kingdom of classification. So good evening, Kamla. So I guess you're new to the session, so welcome. And we'll enter into the kingdom Monera because each of the kingdom has its own unique features. So we can't just hurry up. So we have to understand each of the things. So that's why it is not a topic that you could cover in just 1 lecture or 2 lecture, but we'll go as slow as possible. So we'll cover the kingdom of classification along with kingdom Monera today. We'll try, okay? Because all that we have to cover is kingdom Monera, Protista, and Fungi, and these 3 are just introduction, right? Okay. So now going forward and talking about why do you need classification. So what is the need for the classification? Why it has to happen? So before you actually study something, you should know why, what, and how, right? So first, make sure you address these three questions. So these 3 questions will let you know what is the agenda, the objective behind making you study this topic. You can ask me, ma'am, biological classification on a layman term. It is just classification. I know this the mole that grows on fungi the mole that goes on bread belongs to the kingdom fungi. I know all the green plants are plant kingdom and all the animals are animal kingdom. I am going to be a doctor tomorrow. So what is the need for me to study? Definitely not. So you can't live in isolation, right? So you have to coexist along with us. So today, if you say the pandemic that is holding us all back, that is shaking the entire world, is your coronavirus. So it is a small microscopic organism. So if you want to study, if you want to find a vaccine for that virus, you need to know the structure of that virus, right? So for that, to know the structure of the virus, you should know what family it belongs to, what type of protein it has. So then you trace back its phylogeny, right? So again, you go back to classification. You study about the classification. You need to know all this to face the real life challenge. So no chapter you can say it's not related to you. So every chapter, even though you're going to become doctors or scientists tomorrow or researchers, every chapter has a role in it, okay? So study from that point of view. So talking about why do we need classification? So if you look at classification now, so today you can understand classification is more scientific, okay? So we classified things to understand the type of functions they do, the structure they have, etc., etc. But your classification started way early. Since the time of civilization or evolution started, the process of classification started. But why did it start? So what made classification commence? So what gave birth to the system of classification? So early man, so what he did was, your early man, for his own need, so on a selfish basis, he wanted to classify organism as to which organism he can use to eat, and which organism he can use as shelter, etc., etc. So on a very simple selfish note, he wanted to classify organism, and that has been carried forward, and finally today we stand with a big volume of scientific classification, okay? So that is how it all started. So that is how your story began. So there were many attempts to classify living organisms, because previously in the lecture of living world, you saw how diverse we are, right? So there are so many things that are around us, so many living things you wouldn't have expected, you wouldn't have dreamt of things like that. So there were so many attempts to classify living things. So the need to use organism for our own use. So this was the main motive behind classification. So that gave birth to scientific classification as well. So Aristotle was the first person to give a more scientific classification in the beginning, okay? Fine. So initially the classification was basically living, non-living, and then what came forth was your prokaryotes, eukaryotes, and so on and so forth. Okay, but this is how the story started. Okay, so in the animal kingdom, when we were talking, okay, so we actually spoke about your classification, right? So in the living world, so kingdom, phylum, etc. So do you remember that order? We were talking about that order, right? So what was that order? So first you had kingdom, right? And then what did you have? So for that you have a mnemonic. So the mnemonic says kids prefer cheese over fried spinach green. Okay, so it's enough if you remember the mnemonics. So that is what we are going to talk about. Okay, so do you remember the classification? Kingdom, phylum, class, etc. So do you remember that? So let me give you that mnemonic. So you remember, right? We saw something like kingdom followed by phylum. Do you remember that classification? So what does that mean? So for that I have a mnemonic. So the mnemonic says kids prefer cheese over fried spinach, kids prefer cheese over fried green spinach. So that is how you remember each of them. So let me write it for you on a clear page. So this is how you remember that order. Kids prefer cheese over fried green spinach. So try to remember this. So if you are a foodie, this should not be tough. So all of us love cheese, right? So then you should not forget. So K, kingdom, phylum, class, order, family, genus and species. So this is the mnemonic for that. Okay, okay, anything you can remember. Okay, so if you say King Peter, so you need to remember about a king. So if you are a foodie and if you love eating, if you're really hungry, you can remember this, right? So all of us love cheese and other fast food when you talk about spinach, right? So remember this synonym. So why I'm telling you this synonym is because you need to remember the classification. So it all started with the kingdom, then it goes down to phylum, then class, then to order, then to family, genus and species. Okay, so now we are talking about kingdom monera. So it is a broader classification under which you will have a lot of members. Okay, fine. Okay, so first, what was the attempt to classification? I told you Aristotle was the first one to classify. Okay, fine. From there, how did it go on? So let's see from there, how did it start? So you have something called as the earlier classification. Who came forward with the earlier classification? So the earlier classification was initially 2 kingdom classification. Okay, then you have 3 kingdom classification, then you have 4 kingdom. Then we go ahead with 5 kingdom. So this 5 kingdom classification is the widely accepted one. Okay, so it was given by R.H. Whittaker. So that is the widely accepted one. And then you have the 6th kingdom or the 3 domain. So we'll talk about each of this classification in detail. So why didn't we stop with 2 kingdom, and then why we went on to 3 kingdom, 4 kingdom? So we'll talk about each of them in detail. Okay. Okay, so first starting with the earlier classification. So your earlier classification basically had 2 kingdom. So what was the 2 kingdom? So the first kingdom was plant and animal. So earlier classification, so as the name says, it was done pretty early. Okay, so it was done way early. So during that time, what they did was they understood that there are a lot of living things around them, but they also understood that there was a need to classify. So what did they do immediately? So they immediately classified all the green plants. All that was green was plants. And apart from that, all the other things were animals. So that was initially the 2 kingdom classification. Okay. And what basis did they do? Presence or absence of cell wall. So all those that had cell wall were plants, and all those that did not have cell wall were animals, right? So that was the earlier classification. That was the 2 kingdom classification. But it had a major drawback. So what was the drawback? You were unable to differentiate between unicellular and multicellular organism. So if you classify everything that had cell wall into plants, even some of your bacteria has cell wall. So how will you differentiate between unicellular and multicellular? So that was the problem. Then you were not able to differentiate between prokaryotes and eukaryotes. Your plant, all your plants are your eukaryotes, but your photosynthetic bacteria are prokaryotes, right? So your plants are eukaryotes and your photosynthetic bacteria are prokaryotes. So you were not able to differentiate between them. Then photosynthetic, non-photosynthetic. So you were not able to distinguish between all of these points. So this was a major drawback. Fine. But who gave this classification? It was Carolus Linnaeus. So he gave this 2 kingdom classification. So talking from the question point of view, what will come if I explain you this slide? So what are the possible questions that can come? So remember, there was something called as 2 kingdom classification, but it was eventually ruled out. But that was the first type of classification. Okay, so you need to remember the 2 kingdom, but you need to also remember what was the 2 kingdom. So plant kingdom, animal kingdom. Okay, so this is the first thing that you need to remember, and that is important. The next thing that you need to remember is who proposed it. You need to remember the person. So that is Carolus Linnaeus. So that will be your second point. So this is talking from the question point of view, right? So when you're making notes, if you're making notes right now, just write down kingdom of classification, write earlier classification, write two kingdom, quickly write the two kingdoms and write the name of the scientist. So at the end of the lecture, you should have a rough note which gives you a flow chart. And the points that you note down are the important points that I will be marking on board. Fine? So you need to remember what are the two kingdoms and who is the author. Fine? Understood? So now going forward to three kingdom. Since your two kingdom classification had a lot of restrictions and drawbacks, so people moved on to three kingdom. They thought, okay, so there should be an intermediate group. It cannot be plants and animals. So there has to be an intermediate group. So let's see what is that intermediate group. So here the scientist name, it is again important. So his name was Ernst Haeckel. So remember, three kingdom classification was given by Ernst Haeckel. So why I'm saying the scientist name is important is because one of the previous year NEET question has a match the following, which matches the kingdom name with the person who proposed it. Okay, so there are high chances that you have questions like this. So you need to remember the person who proposed the concept and what was the kingdom of classification. Okay, so what were the three kingdoms here? Kingdom Protista, Plantae and Animalia. Okay, so it is PPA. So that is how you remember Kingdom Protista, Plantae and Animalia. So that was the three kingdom. Fine. So what was included under Protista? So Protista is the new one here, right? So you know plants and animals. So what was included in Protista? So living organisms which do not have tissues. So we eukaryotes have cells. Cells combine to form tissues. Tissues combine to form organs and organs form organ system, right? So such a kind of division of labor or such a kind of organization is not present in certain living organisms. So those were placed under Protista. Fine? Okay. So the first tissue origin in the animal kingdom. So now when I say all the living organisms that did not have tissue origin were placed in Protista, which means your plant and animal kingdom had tissue structure, right? So the first tissue origin in animal kingdom was your coelenterata. Okay. The second one, in case of your plant kingdom, it is Bryophyta. So please remember these examples. They are exceptional examples. Okay. So what did the kingdom Protista include? So on a whole, it included your prokaryotes, protozoa, porifera, algae and fungi. Okay. So these statements are important because when you observe my slides, I will not have a lot of theory. So when I have a lot of theory, it means these are important. You'll have questions like which of the following statement is incorrect. Okay, so these are the questions which are very tricky in your NEET exam. So you need to understand each of the statement and you cannot go wrong. So for that, sometimes you'll get statements like this. So kingdom Protista in the three kingdom classification includes what? So you need to know each of the members, right? So it is your prokaryotes, protozoa, porifera, algae and fungi. Fine? Okay. So this is your three kingdom classification. Remember the name of the scientist and what were the three kingdoms. Okay, so this is point 2. Fine? Now coming to four kingdom classification. So four kingdom classification was proposed by Copeland. So again, he is the name of your scientist. So you need to remember this. Fine. So Munira. So the fourth kingdom. So three kingdoms you know. Yes, Priyanka. So Copeland gave us your four kingdom classification. So Protista, Plantae, Animalia you know. So what was the new addition here? That was your Munira. So Munira contained all your prokaryotes. So prokaryotes include your bacteria, mycoplasma, blue-green algae, etc. Fine. Your Protista included all your eukaryotes. So basically your unicellular eukaryotes. Fine. Then you have Plantae. So remaining eukaryotic plants. Then animals will have the remaining eukaryotic animals. So this was about the four kingdom classification. If you ask me why the three kingdom classification was a failure, so can you tell me why it was a failure? So since it was a failure, we moved on to the fourth kingdom of classification because people were not satisfied in putting all your prokaryotes and eukaryotes combined together in Protista. So there are a lot of prokaryotes and eukaryotes without tissue origin. So you cannot couple everything into kingdom kingdom Protista. Hence we came forward with the four kingdom classification which included Munira. Fine. So who proposed it? Copeland. So you should know what are the four kingdoms. Fine. So finally, People were not satisfied with the 4-kingdom classification as well, because something was again missing. So people had a lot of contradiction. Okay, now 4-kingdom classification also includes a lot of eukaryotes and prokaryotes together. So people were not clear with the distinction of protista and plant kingdom, because some unicellular eukaryotes also had photosynthetic ability. So because one which has photosynthetic ability, we call it plants, right? But they also found some unicellular eukaryotes which were capable of doing photosynthesis. Now they were pretty confused. So how to classify them? Then we have a brilliant classification, that is your 5-kingdom classification. So today your biological classification, when I say kingdom Monera, Protista, so that is based on your 5-kingdom classification. So this is the widely accepted one. Fine. So this was given by R.H. Whittaker. Again, the name of the scientist is important. Okay. So it is based on 5 features. He considered 5 main features and he classified based on them. So your first feature is your cell structure. So how is the cell? So what is the type of cell? Whether it has a cell wall or not. Okay. The second one, thallus organization. So thallus means the body organization. So what type of complexity? Whether it is multicellular, unicellular, etc. Fine. The third one is nutrition. So it can be either autotrophic or heterotrophic. Fourth one is reproduction, the ability of it to reproduce asexually or sexually. Fifth one is phylogeny. So what do you mean by phylogeny? What do you mean by phylogeny? It's evolutionary relationship. How closely it is related to the other kingdom. How closely it is related to the other organism. So that is called as phylogeny. Fine. So based on these 5 features, your R.H. Whittaker gave us the 5-kingdom of classification. Okay. So he was Robert, so R.H. stands for Robert Harding Whittaker. So he gave us the 5-kingdom of classification, and this is the most widely accepted. Okay, and we are talking about this 5-kingdom of classification now. So the examples of 5-kingdom. So just relax, don't panic looking at the examples. It is fine. I am just introducing the examples. So as we talk about them, each of them in detail, so you will become familiar. Okay. So your Monera, Protista, Mycota. Mycota is your fungi. So the 5th kingdom was your fungal kingdom. Okay, the 5th kingdom that was introduced was your fungal kingdom. So Monera was already there, Protista was already there, animal kingdom, plant kingdom were also there. So the 5th kingdom that was introduced was your fungal kingdom. Okay, why Porifera was included in Protista in 3 kingdom? So what does Porifera mean? So basically they classified that, okay, so plants are the green plants, so they are eukaryotes. So animals were the eukaryotic animals that were included. So in the 3-kingdom classification, the other only kingdom that was present was Protista. So apart from all the eukaryotic plants and animals, all the other organisms were put into kingdom Protista. It was not a proper classification, and that's why it was a limiting factor. That's why they move forward with your 4-kingdom classification. Okay, Rajesh? Fine. So this is your 5-kingdom. So you have your kingdom Protista, Monera, fungi, plants and animals. Okay. So your protistans are basically your unicellular eukaryotes. Okay. Why it was called true fungi? Okay. So what do you mean by true fungi is sometimes you have something called as it is fungal like, it is bacterial like, but it is not exactly. So when I say true fungi, it is very distinct from all the other organisms, and it actually belongs to a very separate kingdom. Okay, so that is why when your fungal kingdom was introduced, we called it Mycota, because the study of fungi is called as mycology, right? So it actually is a true fungi, meaning it is a very distinct structure on its own. Okay, it does not resemble either of the kingdom. It does not resemble your protistans, nor it resembles your plant kingdom. Okay, so it is one unique kingdom. So though 5-kingdom of classification was widely accepted, and today we are studying 5-kingdom of classification, still there was another scientist who was called as Carl Woese. Okay, I guess I have the name. Yes, he said no, it is not 5-kingdom of classification. I don't agree. It has to be 6-kingdom of classification. Already from 2-kingdom to 5-kingdom, it is pretty complex. Now he say no, still 5-kingdom of classification does not satisfy all the criteria, because in kingdom Monera you have classified. So in kingdom Monera you have 2 groups. Okay, so kingdom Monera is basically your prokaryotes. So kingdom Monera has 2 basic classes, that is Archaebacteria and Eubacteria. He said these 2 bacteria itself belong to 2 different kingdoms, so divide them further. So your 5-kingdom was further split into 6-kingdom. Okay, so your Archaebacteria and Eubacteria, they belong to kingdom Monera. So your Carl Wu said that no, your archaebacteria and your eubacteria are way different. They cannot come under the same kingdom, so further separate them. So he made it totally 6 kingdom. But later he analyzed and observed his result, and he came up with a conclusion saying, no, it is not 6 kingdom, but only 3 main domains that are present. So what were the 3 main domains? Okay, so initially why did he say archaebacteria and eubacteria have to be separated? So he had a valid point. He said it is because of the absence of peptidoglycan in the cell walls of archaebacteria, which is unlike those in eubacteria. Fine, so that was the reason he gave. But then he himself did an analysis and concluded that there are only 3 main domains and not 6 kingdom of classification. So on what basis did he give this conclusion? Was when he studied the 16S ribosomal RNA gene. So this 16S ribosomal RNA is present in the 30S subunit of your ribosome. So you remember us studying the ribosome, right? Yes, in your prokaryotic ribosome, it is 30S and your 70S. So your 30S ribosome contains this 16S ribosomal gene. Ribosomal RNA. So what is the unique feature of this RNA is that this RNA has a very slow rate of evolution. When I say a very slow rate of revolution, so it remains the same for a long period of time. Over million years also, this 16S RNA will not get mutated, will not have any variations. So it has a very slow rate of mutation, hence it can be used to construct phylogeny. So it was easy for us to study the 16S RNA and conclude which came first and what basis you have to classify them. Hence, on the basis of your 16S ribosomal RNA, he classified saying there are not 6 kingdoms but only 3 main domains. So the 3 main domains were your archaea, bacteria and eubacteria. Okay, so your archaea, bacteria, eukarya. So these were the 3 main domains. So he said your bacteria had kingdom eubacteria, archaea had kingdom archaebacteria, and then your eukaryote had plants, animals, fungi and protista. So this is what he concluded. Since he accepted that there are only 3 main domains, the widely accepted system of classification was your 5 kingdom of classification. Okay, so we went forward with your 5 kingdom of classification. So today we will talk about your 5 kingdom of classification and we will begin with kingdom Monera. Fine? Okay, so welcome to kingdom Monera. So these microorganisms, though they are small, they are pretty beautiful, and always the small things make big difference, right? So no one ever cared about a microorganism before, but today we understood microorganism can be as worse as a bioweapon. Okay, so it can create something called as a bio-war, right? So that is what the initial talks during the pandemic, right? Okay, so this COVID-19 was a bio-war, it was a bioweapon used by China, etc., etc. So I'm just telling this to say, though they are microscopic, they have a very high potential, and it is always the small things that make huge difference. But apart from all that, your microorganisms are beautiful creatures. So when you observe them under the microscope, they are very, very beautiful. Okay, so from the time you wake up till the time you go to bed, and even after you sleep, you are living with microorganisms. Okay, you might live alone in your house, you might have a home alone day, but still, from the time you wake up, from the time you brush till the time you go back to bed again, so you are living with microbes. So everywhere, so say for example your saliva has microorganism, the food you eat, suppose if you are taking idli, so how is the batter fermented? Again a microorganism plays a role, right? So again you eat food, okay, so you have digestive enzymes and you have what is called as bacteria, the gut microbiome enabling your digestive process, everything. So if you take curd, again what turns milk into curd? Again it is a bacteria. So a life is a whole cycle filled with microorganisms. Without microorganisms, neither you nor I can exist, right? So we are all living with microorganisms. You and I are not sterile. So maybe you can wash your hands 10,000 times, but still you'll have 1 bacteria. Okay, you can wash your hands, but what about your stomach? If you make your stomach sterile, you won't be able to survive. You need microorganisms for the proper functioning of your body, right? So microorganisms are actually important. So we will talk about the role of microorganisms in your chapter in class 12. But why I am saying this is kingdom Monera is basically a prokaryotes. So you'll be amazed at how diverse they are, how beautiful they can function, okay? So they can live everywhere, okay? So extremely everywhere. So like we spoke about in the living world. So let's go forward and see what we are going to talk about in kingdom Monera. So we'll talk about the main characteristics of Monerans. So we'll talk about archaebacteria, eubacteria, mycoplasma, actinomycetes, cyanobacteria. Okay. So these are the members of your monerans. Fine, but how do you remember them? So they are pretty complicated. So you can remember them in any order. So for example, I made a statement saying an episode of comic makes you feel really amazing. Okay, so there you have all the points done. Okay, an episode of comic makes you feel really amazing. So that gives you a mnemonic for all the members of your monerans. Okay, so if you want, you can note down. Okay, so you can say an episode of comic makes you feel amazing or makes you feel really amazing. So the R there stands for rickettse. Okay, so that is also one way you can remember. So understand the main classification of your kingdom Monera is archaebacteria and eubacteria. Okay, so they are the main members. But apart from that, you have other members like mycoplasma, actinomycetes, okay, and cyanobacteria. Okay, so there are the other members. So people usually tend to get confused between whether they all belong to archaebacteria or eubacteria, because when we were talking about the sixth kingdom of classification, I told you your archaebacteria and eubacteria are the main ones in kingdom Monera. But no, they are the main ones, but apart from that, you have other members as well. So we'll talk about each of the members in detail. Okay, so good evening. Thank you, Suraj. Fine. So going forward, so general characteristics about your kingdom Monera. So we are talking about your monerans here. So they're all prokaryotes. So when I say prokaryotes, they're all unicellular here. Fine. So they're the most primitive forms of life. They have been originating from the ancient livestock. So they are known as progenate. So what do you mean by progenate? So you can call them as the last universal ancestor available. So you go back, right? So when you look at your family, so your grandparents, great-grandparents, great-great. So finally you have one end, right? So that end for all living organism is your monerans, because they are your progenate. So they are called as the last universal ancestor. So that is what progenote mean. Okay, so the last universal ancestor. Fine. So we are going to talk about the various types of monerans here. So the main characteristic, so the cell type is prokaryote. Yes, so all your prokaryotes come under kingdom Monera. So the cell wall is not cellulosic, okay, but it is made up of polysaccharides. So PS stands for polysaccharides and AA stands for amino acids. Fine. So nuclear membrane here is absent. Okay, so they do not have a nuclear membrane. So body organization, it is cellular, but your membrane bound organelles are absent. Fine, because they are prokaryotes. Your prokaryotes do not have membrane bound organelles. Nutrition, so what is the mode of nutrition? They can be autotrophic, they can be heterotrophic. Heterotrophic includes saprophytic and parasitic. So when I say autotrophic, it means they can prepare their own food. So that autotrophic is also divided into two types. It can either be photosynthetic or chemosynthetic. So when they depend on your light energy, it is called photosynthetic. When they depend on your chemical and other energy, it is photosynthetic. Fine. So heterotrophic. So when I say saprophytic, they feed on dead organic matter. Parasitic, they are going to be harmful for the host. Fine. Okay. So examples of your Moneran kingdom. So we have made a mnemonic as well. So this is what you have to remember. Okay. Fine. Okay. So the first one, we are going to talk about your archaebacteria. So your archaebacteria, they are believed to have evolved immediately after the origin of life on Earth. So I said your kingdom Monerans were the primitive organisms on Earth. So among the monerans, your archaebacteria has the privilege. So they have arrived on Earth since the time life on Earth was existing. Okay, so they can live under extremely diverse conditions. Okay, so they can live in very high temperatures like hot springs. So the temperature is approximately 80 degrees here. It can have high salt. It can also survive in high salt concentrations. So that is called as salt marshes. So imagine high salt concentration. The pH is more saline. So they are able to survive in extremely saline conditions as well as in extremely acidic conditions as well. Fine. So they are also known as living fossils. So your archaebacteria has the privilege of being known as a living fossil. So what do I mean by living fossil? So for the long time, from the time they existed on this planet Earth till now, they are in the same life form. They have not evolved, they have not mutated, nothing. Okay, so they're just the same. So they have existed in this earth for over millions of years. So they are called as the living fossils. Fine. So they are generally anaerobes. So this condition is pretty important. You need to remember. So your archaebacteria, they are generally anaerobes. Fine? Okay. So going forward, so how is the cell designed? So what is the architecture of the cell? The cell wall is made up of polysaccharides and polypeptides. So PS stands for your polysaccharide. Fine? Okay. Your cell membrane, it is a single layer of branched chain molecules of lipids. So your cell membrane is made up of lipids. So remember that much. And this lipid is a branched chain. Okay. So since they have a branched chain made of lipid molecules, they are very resistant. So it is not easy for any drug to enter. This is also one reason that your bacteria are drug resistant initially, but not now. Now we know the mechanism. So they have a single layer of branched chain molecules which are made of lipids, and thus makes them more resistant. Fine. So they have introns in DNA. So what were introns? So introns were intertwining sequences, right? So you have a single segment of DNA. So a segment of DNA is called as a gene, which codes for a protein, right? So for example, if this is a gene and this gene has sequences like this. So this portion codes for a protein, but these two do not code for a protein. So they are just junk sequences in between coding sequences. So these are called as introns. Fine. So they have introns present in them. So ribosomal proteins are present because ribosomes are present in both prokaryotes and eukaryotes. Okay, so they are highly acidic. And they have histone proteins as well. So histone proteins help in packing your DNA. Okay, so they help in compacting your DNA. Fine. So this is your general design of your monomerans. Now going forward and talking about the special kinds of monomerans, because archaebacteria, because they can survive in extreme condition. So they are pretty different. The first one is methanogens. So methanogens means the methane producing bacteria. Okay, so they can produce methane. So they convert carbon dioxide into methane. Fine. So what do? They convert organic substance such as cellulose into methane. So it is also called as the Gobar gas fermentation. Okay. Fine. Okay. So one example. So examples are pretty important. Again, you have questions like match. So that is where you tend to get tricky and lose points. So methanogens, remember methanobacterium or methanococcus. So anything that starts with methano is your methane producing bacteria. So just as the name says methanogens, so you can just add bacterium to it or coccus to it. So methanobacterium or methanococcus. Fine. So this is easy to remember. I don't think you have any problem in remembering this. Fine. Since it acts on a cellulosic substrate, so there are high chances that it can also be found in the gut of your rumen. In the rumen of your cattle. Because your cattle feeds on a lot of plants, right? They're herbivores. So the plants have a lot of cellulosic content, but you need someone to act on this cellulosic content. Whereas human beings cannot digest a lot of cellulose, right? So that's why we ensure our food is cooked properly. So a lot of cellulose is not digestible by us. So in bacteria, in case of rumen, so they consume a lot of cellulose substance. So your methanogens will act on them and help it to break into a simpler substance. Fine. So that is the role of methanogens. So when it is found in the rumen of cattle, it is called as rumenococcus. So coccus refers to the shape of bacteria. Fine. So that is rumenococcus. Okay. So going on to the next type, that is halophiles. So halophiles are the ones that can survive in extreme saline conditions. Okay, extremely saline habitats. So example, so they are under the broad term called halophiles. So remember, they are halobacterium. Fine. So there it was methanobacterium and methanococcus. So here you combine both the things, halobacterium, halococcus. So this put together is one name. So it is the binomial nomenclature for one bacteria. Fine. So halophiles, so what actually happens, how are they able to survive in saline condition is that they have a purple membrane around them, and this purple membrane absorbs bright light and directly converts them into ATP. So that is how they are surviving. So they cannot prepare food on their own. So they are non-photosynthetic. They just have a purple membrane which directly produces ATP molecules. Fine. For example, when you have a tide like this, which is purple in color, okay. So this purple in color tide is because of the presence of the purple membrane, that is your halophiles. Fine. Is that understood? Okay. So now going forward, so we'll talk about the next important bacteria. Your archaebacteria, that is your thermoacidophiles. So thermoacidophiles. Thermo is high temperature, acido is acidic condition, and philes means loving. So the bacteria that loves to survive in high temperature, high acidic condition. Okay, so they are chemoautotrophs. So they depend on the chemical substances for preparing their food. So that is called as chemoautotrophs. Fine. So they are found in very extremely high temperature, that is around 80 degree. So they can survive in acidic pH as low as pH 2. For example, in hot sulfur springs, they are also found. So what do they do there? How do they act? So I said they are chemoautotroph. So they have to use the chemical substance, right? So they oxidize sulfur to H2SO4 to produce energy, and that energy is used to produce food. So they obtain their energy from a chemical reaction. So it is chemoautotroph. Fine. So they are generally aerobes. Fine. Okay. So they are exceptionally aerobes. Example is Thermoaquaticus, so sulfobolus and thermoplasma. Okay. So is this understood? Fine. So this is about thermoacidophiles. Now going to the next class. So we are done with Archaebacteria. Now we are entering into Eubacteria. So we are done with the first member of your Muniran kingdom. Now coming to the second one, that is Eubacteria. Bacteria are cosmopolitan. So what do you mean by cosmopolitan? They are present everywhere, right? So they are omnipotent. So omnipresent you say. So everywhere there is bacteria. So when I say bacteria is there everywhere, on your palm, on the screen, etc., etc. So that is the Eubacteria that is present. So Archaebacteria are the one that can survive in extreme conditions. So you won't find a thermoacidophilus bacteria right here, right? So it cannot survive in this condition. It has the ability to survive in that particular condition. So when I say bacteria is cosmopolitan, so that refers to Eubacteria. So they are present everywhere. Fine? Yes, everywhere, literally everywhere. Okay. So how is the design and structure of them? So they have what is called as a capsule, glycocalyx or slime mold. So that is how their envelope is designed. Fine? Okay. So the bacteria has a capsule. So when the bacteria has a capsule, it is called as a capsulated bacteria, and capsulated bacteria makes it very virulent or pathogenic. Okay, so your bacteria without a capsule is non-virulent, but a bacteria with a capsule is virulent. Okay, so I'm sorry for the spelling of bacteria. Fine. So now the next point. So the capsule can be made up of a polysaccharide and a polypeptide. So that protects them from WBCs. So when a bacteria enters your body, when a bacteria enters your body, it is foreign to your body. It is pathogenic to your body. So your body's first line of defense will try to attack that bacteria. But when your bacteria is capsulated, it is way smart that it escapes from your first line of defense. Okay, so that's why it becomes pathogenic and virulent. Fine. So the next one is cell wall. The cell wall is made up of peptidoglycan or murin. So it is also known as mucopeptide. Fine. Okay. So your polysaccharides here, they can be of your glucose, mannose, galactose, amino acid, sugar units. Fine. So good evening. So amino sugars, so N-acetyl glucosamine and N-acetyl muramic acid. So they are the two amino sugars present on your cell envelope. Okay. So they are just the components that I am talking about. So your cell wall is made up of peptidoglycan. You need to remember it is also or peptidoglycan or murin. So you need to remember these two terms. Fine. Then there is something called as amino sugars that connects your cell envelope, something like this. So you have a NAG and NAM. Okay. So this is N-acetyl glucosamine and this is N-acetyl muramic acid. Fine. Okay. So this is how your bacterial cell wall is designed. So no doubts, right? So till this no doubts. So can we go forward? So we are almost done with your Archaebacteria. We are entering your Eubacteria. So I believe there are no doubts. So going forward, let's talk about gram-positive and gram-negative bacteria. So under your Eubacteria, you have this classification of gram-positive and gram-negative bacteria. So why does this classification arise? On what basis does this come? So look at this figure. So here it talks about your gram-positive bacteria and here it says, gram-positive and here it is gram-negative. So when you look at the design or when you look at the cell wall of these two, there's a pretty good difference, right? So your gram-positive bacteria has a thick peptidoglycan layer. It's a pretty thick layer, right? Followed by your lipid layer. So it is just two layers, one thick layer of peptidoglycan and one small layer of your lipid. Whereas your gram-negative bacteria has two lipid layers and sandwiched peptidoglycan layer, right? So it has a lipid bilayer. In between you have a peptidoglycan layer. So on this basis, it has been classified as gram positive, gram negative. But you can ask me, ma'am, the name does not coincide. So what does gram mean? So gram was the name of a person. So he was Hans Gram. So he helped with this classification. But what actually it means is that when you add a stain to this bacteria, so you have two types of bacteria, you add a particular stain. For example, the stain is crystal violet. Okay. So both your bacteria will observe the stain will enter inside your cell. Okay. After some time, when you wash the stain, okay, on top of it you wash, so the stain is washed off. But you will when you observe under the microscope, you will be able to understand that your gram positive bacteria is able to retain the stain. For example, let's say you apply a tattoo or a mehendi on your hand and then you wash it, but still the color of the mehendi is retained, right? Something like that. Okay, your bacteria is able to retain, gram positive bacteria is able to retain the strain, whereas your gram negative bacteria will not be able to retain the strain. So this is able to retain the strain because of the thick peptidoglycan layer. Fine. So on this basis, it was classified as gram positive and gram negative. Does gram negative bacteria have two cell membrane? Not two cell membrane, it has two lipid layers. So we don't say it has two cell membrane, but we say it has two lipid layers and a peptidoglycan layer sandwiched in between. Fine. Is that understood? Okay. So clinically, when you talk about, so when you want to classify, because we have a lot of bacterial infections affecting humans, right? So when you want to classify it as gram positive, gram negative bacteria, so it is a type of clinical diagnosis. So when you want to kill your gram positive bacteria with a drug, the mechanism is different because it has to penetrate a thick peptidoglycan layer. When you're talking about a gram negative bacteria, the mechanism for the drug to kill this bacteria will be different because the drug has to enter inside, right? So it also has a clinical application. So not all your gram positive bacteria are harmful and pathogenic, but your gram negative bacteria, almost all your gram negative bacteria are pathogenic. Fine? Is this understood? So I think we will stop here because let's not go forward. We still have to talk about your cyanobacteria, actinomyces, but that's still fine. We'll cover it in the next chapter because today I just want to introduce to you what is biological classification. I don't want you to get saturated and run away. So we'll stop right here, and it's enough if you revise this concept, because in the next class we will speed up and talk about, we'll finish your kingdom Munira and then go forward with your kingdom Protista. Fine. So till this, was it clear? So do you remember the mnemonics that I told you? So now can you easily give me an example of eubacteria, archaebacteria? Eubacteria can be any bacteria. Your E. coli is a eubacteria, but for archaebacteria you can give methanobacterium, etc. Right? So the examples are very important. So the more you remember every day, the more easier it is. So one trick that I can tell you, if you actually want to remember things, so when you go to bed, and the time you go to bed and the time you fall asleep, so there's a gap in between them, right? So no one just falls on bed and falls asleep, right? So you twist and turn, you just think of all the things and then you casually close your eyes. So that period, that intermittent period, the time when you go to bed and the time you fall asleep, is a very, very precious time. So try recollecting whatever you learnt, it will stay in your memory forever. So similarly, when you wake up in the morning, the moment you open your eyes and the moment you get out from bed are different, right? So there's a period of interval between them. No one opens your eyes and walks out of bed, right? Unless and until you are in a hurry. So the moment you wake up until the time you actually consciously start working, so that time is also very precious and your brain is very active. So try recollecting all the names, you will never forget. So do this for 1 week. So these names will stay in your brain forever. Okay, so that is one trick. So try recollecting whatever you did during the day. So your memory power will also be enriched and you'll also remember the examples. Okay, so you don't have to remember the examples of biological classification all your life. At least till your exam you have to remember, right? So try this. So every day. So whatever you feel tough, just keep remembering it. So that is easy. Fine? Okay, so we'll stop with this. So if you really like, if you genuinely understood something out from this lecture of 45 minutes, so please do subscribe, like and share. Fine? So I will meet you on Monday. So on Monday we'll be talking about a class 12 chapter. But again on Tuesday we'll be back on track talking about the sneak peek into the world of biological classification. So until then, have a happy weekend. So relax, enjoy, but at the same time do study. So everything is equally important. Fine? So until then, it is me signing off and thank you.